I am Dr. Nikita Sharma, Associate Professor Chemistry, SPNKS Government PG College, Dausa. Today we will start biotechnological applications of enzymes. Enzymes are essential components of animals, plants, and microorganisms due to the fact that they catalyze and coordinate the complex reactions of cellular metabolism. Up until the 1970s, most of the commercial application of enzymes involved animal and plant sources. At that time, bulk enzymes were generally only used within the food processing industry, and enzymes from animals and plants were preferred as they were considered to be free from the problems of toxicity and contamination that were associated with enzymes of microbial origin. However, as demand grew and as fermentation technology developed, the competitive cost of microbial enzymes was recognized and they became more widely used. Economic advantages. The sheer quantity of enzyme that can be produced within a short time and in a small production facility greatly favors the use of microorganisms. For example, during the production of rennin, a milk coagulating enzyme used in cheese manufacture, the traditional approach is to use the enzyme extracted from the stomach of a calf, a young cow still feeding on its mother's milk. The average quantity of rennet extracted from a calf's stomach is 10 kg and it takes several months of intensive farming to produce a calf. In comparison, a 1000 liter fermenter of recombinant Bacillus subtilis can produce 20 kg of enzyme within 20 hours. Thus, the microbial product is clearly preferable economically and is free from the ethical issues that surround the use of animals. Indeed, most of the cheese now sold in supermarket is made from milk coagulated with microbial enzyme, so is suitable for vegetarians. A further advantage of using microbial enzyme is their ease of extraction. Many of the microbial enzymes used in biotechnological processes are secreted extracellularly, which greatly simplifies their extraction and purification. Microbial intracellular enzymes are also often easier to obtain than the equivalent animal or plant enzymes as they generally require fewer extraction and purification steps. Animal and plant sources usually need to be transported to the extraction facility, whereas when microorganisms are used, the same facility can generally be employed for production and extraction. In addition, commercially important animal and plant enzymes are often located within only one organ or tissue, so the remaining material is essentially a waste product, disposal of which is required. Finally, enzymes from plant and animal sources show wide variation in yield and may only be available at certain times of year, whereas none of these problems are associated with microbial enzymes. Technical advantages. Microbial enzymes often have properties that make them more suitable for commercial exploitation. In comparison with enzymes from animal and plant sources, the stability of microbial enzymes is usually high. For example, the high temperature stability of enzymes from thermophilic microorganisms is often useful when the process must operate at high temperatures during starch processing. Microorganisms are also very amenable to genetic modification to produce novel or altered enzymes using relatively simple methods such as plasmid insertion. The genetic manipulation of animals and plants is technically much more difficult, is more expensive, and is still the subject of significant ethical concern, especially in the UK. Enzymes may be intracellular or extracellular. Although many enzymes are retained within the cell and may be located in specific subcellular compartments, others are released into the surrounding environment. The majority of enzymes in industrial use are extracellular proteins from either fungal sources, Aspergillus species, or bacterial sources, for example, Bacillus species. Example of these include alpha amylase, cellulase, dextranase, proteases, and amyloglycosidase. Many other enzymes for non-industrial use are intracellular and are produced in much smaller amounts by the cell. Examples of these include asperginase, catalase, cholesterol oxidase, glucose oxidase, and glucose 6-phosphate dehydrogenase. Enzyme purification. Within the cell, enzymes are generally found along with other proteins, nucleic acids, polysaccharides, and lipids. The activity of the enzyme in relation to the total protein present, that is the specific activity, can be determined and used as a measure of enzyme purity. A variety of methods can be used to remove contaminating material in order to purify the enzyme and increase its specific activity. Enzymes that are used as diagnostic reagents and in clinical therapeutics are normally prepared to a high degree of purity because great emphasis is placed on the specificity of the reaction that is being catalyzed. Clearly, the higher the level of purification, the greater the cost of enzyme production. In the ease of many bulk industrial enzymes, the degree of purification is less important, and such enzymes may often be sold as very crude preparation of culture broth containing the growth medium organism, whole or fermented, and enzymes of interest. However, even when the cheapest bulk enzymes are utilized, example proteases for use in washing powder, the enzyme cost can be contributed around 5 to 10% of the final product value.